Hey there, listeners. Welcome to another paper preview podcast. Credit for this work goes to the original authors. Please find a link to their paper on PubMed Central in the description below or using the QR code on screen. This paper preview is meant to be a general overview and introduction to the paper. Please read the actual text and figures for more details. The following discussion, script, and voices were created using AI and are not intended to mimic the likeness of the original authors. As with any use of AI, all factual information should be double-checked with the original source.
Okay, let's unpack this. Imagine your cells, right, particularly your neurons, actively throwing out their garbage. And I don't mean just a few rogue molecules. I'm talking about, large visible clumps of misfolded proteins, even entire damaged organelles, literally ejecting them. Now, for any of you deeply immersed in biomedical research, this immediately brings to mind the immense challenges of neurodegenerative diseases. We're all, acutely aware of how critical misfolded proteins and mitochondrial dysfunction are in neuronal decline. And we know just how much cellular machinery is already dedicated to maintaining health through proteostasis, chaperones, autophagy, the proteasome, mitophagy, the whole works. But what if there was another previously unrecognized large-scale cleanup mechanism? Today, we're diving deep into a truly groundbreaking paper, C. elegans, neurons, jettison, protein aggregates, and mitochondria under neurotoxic stress, published in Nature. It reveals an astonishing and frankly entirely new mechanism. It could fundamentally change how we approach these diseases, how we think about cellular waste management, and we are incredibly fortunate today. We have one of the paper's lead authors and the corresponding author with us who can walk us through this incredible discovery. As editor, I can tell you when this paper came across my desk, it was immediately clear this was something special, a real paradigm shifter. So our mission today is to explore this novel pathway in detail, understand its mechanics, its deep implications, and really consider what this means for our understanding of neuronal health and disease, especially for those of you pursuing biomedical research in this very field.
Thank you so much for having me. It's truly a privilege to discuss this work and share the journey behind discovering X-verse. Getting this published in Nature was, a huge milestone for us, obviously.
Yeah.
And I'm always really eager to engage with aspiring researchers about its potential.
Absolutely. And it's our privilege to delve into such a pivotal finding. When a paper like this lands, the first question is always, how did they even find this?
Yeah.
So let's start right at the very beginning. What was that initial spark, the first observation that hinted at this, extraordinary phenomenon? Where did this journey begin for your team?
Well, it's funny. The discovery of X-verse was, in many ways, entirely serendipitous. Really. We weren't actually setting out to find a new cellular garbage disposal system at all.
Mm-hmm.
Our lab was focused on something else entirely, actually. We were studying age-associated dendritic restructuring in C. elegans, neurons, how the branches change shape as the worms get older.
Yeah.
So we were using specific fluorescent reporters, things like GFP and M-cherry fusions, expressed in neurons like the ALMR mechanosensory neurons. These let us visualize their shape, track changes over time as the animals aged. We had these meticulous time-lapse imaging setups. We'd watch these worms for hours sometimes, believe it or not, even days, on these little agar pads under high-resolution fluorescence microscope.
Right, painstaking work.
Exactly. And what started to appear, just intermittently at first, wasn't the kind of changes we expected within the dendrites. It was these distinct, intensely fluorescent blobs. And the key thing was they were consistently outside the cell membrane. But you could clearly see they originated from the neuron. So our immediate thought, as I think any good scientist would be, was skepticism, pure skepticism.
Mm-hmm.
Is this an artifact? Is the neuron bursting? Is it something we're doing with the imaging, maybe phototoxicity or mechanical stress from mounting them?
Sure, you have to rule all that out first.
Absolutely. We went through a really rigorous process of elimination, checking for signs of cell damage, varying the laser intensity, trying different mounting conditions, using different fluorescent markers, everything we could think of. But these structures, they persisted. And crucially, they looked too organized to be just random debris. They were clearly membrane-bound, often quite spherical. And we could actually watch them slowly detaching, moving away from the soma of the cell body. That's when the real detective work began, trying to figure out what this organized extrusion actually was. And we decided to coin a term for them, X-verse, for their external cellular origin.
That sounds like a true scientific detective story. I can just imagine the team meetings, the debates. Is it real? Is it an artifact? Was there a particular moment, maybe a specific image or video that finally shifted the team's view from artifact to, okay, this is a real discovery?
Oh, definitely. Yeah, there was one day, I remember it so clearly. We're looking at an ALMR neuron doing a time lapse. We'd been worried, as I said, about pressure, maybe the mounting media causing stress, leading to these blebs. But then we saw this one structure. It was slowly forming, bulging out from the soma, almost like inflating a tiny balloon. And it wasn't just, blebbing passively. It was actively accumulating the fluorescently tagged proteins inside it, specifically the aggregation-prone ones we were tracking. And then right there on the screen, we watched it pinch off and just drift away. It wasn't explosive, not chaotic like a cell bursting. It was a relatively slow, almost deliberate-looking process. We timed it anywhere from maybe 15 minutes to, well, sometimes over an hour and a half, from the initial bulge to complete detachment. Seeing that whole process, the biogenesis in real time, and seeing its specific cargo, that was the moment. That's when we knew we were seeing something purposeful. It stopped being just an anomaly, it became a phenomenon that was just begging for an explanation.
That kind of detailed observation, seeing the whole process unfold, that's really powerful for establishing something entirely new, especially something so visually striking. It really helps separate these structures from just, cellular junk.
Okay, so you've clearly defined these structures. What was the next big question then? How did you go from, okay, they exist, to suspecting they were this purposeful mechanism for clearing out cellular burdens?
Right, that's the critical next step, isn't it? Because a novel observation, well, it's just that until you demonstrate its biological significance, the initial key really was their sheer physical characteristics. Exifers are remarkably large. We measured them, and they averaged around 3.8 micrometers in diameter. Now, to put that in perspective for listeners, that's comparable in size to an entire neuronal cell body, the soma, in C. elegans.
Wow, okay. That's huge compared to other vesicles. Exactly. It immediately set them apart from other known extracellular vesicles, things like exosomes, which are tiny, 30 to 100 nanometers, or even migrosomes, which are generally smaller and have a totally different way of forming. Their size alone was a clear distinguishing feature. It strongly suggested they could encapsulate a really substantial amount of cellular material.
More than just a few molecules.
Way more. And beyond the size, their biogenesis, the way they formed, was unique. We spent, countless hours with time-lapse microscopy recording how they form. And it's this really striking process. The neuron soma develops this balloon-like extrusion, as we called it. Often you see it accumulating fluorescently labeled protein. This bulge then gradually pinches off from the cell. Sometimes it stays connected for a while by this very thin, thread-like tube, almost like an umbilical cord. We even saw instances where this thin connection seemed to facilitate the transfer of tagged proteins and even calcium signals into the attached exifer compartment.
Really? So it's still connected functionally for a time?
It appears so, yeah. Suggesting a dynamic, maybe even regulated link before that final disconnection. This wasn't some random rupture. It looked like a structured multi-stage event. And of course, we had to rigorously rule out that these weren't just cells dividing or maybe undergoing some weird form of cell death. So we confirmed when DPI staining exifers do not contain nuclear DNA, unlike a dividing cell. The neuronal somas, meanwhile, remained DPI positive. Clear difference. Furthermore, when we treated the worms with cell division inhibitors like hydroxyurea or used RNAi against key cell cycle genes, it had no effect on exifer levels.
Okay, so definitely not cell division.
Definitely not. That firmly established exifergenesis as distinct from classical cell division. We also use plasma membrane reporters specifically, PMEC4pH, PROSI delta, GFP, and confirmed with electron microscopy that these structures are indeed enclosed by a continuous membrane. So they are true vesicles, not just random blebs of cytoplasm spilling out.
Got it. Membrane-bound, large, unique formation.
Exactly. And what also pointed towards their significance was finding them across various neuronal types in C. elegans. While we first saw them with specific fluorescent reporters, we quickly checked they weren't just an artifact of the transgene expression. We saw them in amphid neurons after dye filling, which means they can form under, native physiological conditions, even without the stress of expressing foreign proteins. We detected them most frequently in those ALMR gentle touch receptor neurons, interestingly, but not in the PLM touch neurons. But they were also present in dopaminergic neurons, the PDE and CEP neurons, MLP neurons, and the sensory acer neurons too. This ubiquity, seeing them in diverse neuronal types, strongly suggested that exophagenesis represents a fundamental conserved cellular process, not just some isolated quirk in one specific cell type. So yeah, the combination of their distinct size, their unique biogenesis being membrane-bound, and their prevalence. It all strongly led us to believe we were looking at something purposeful and biologically significant, a real cellular maintenance mechanism.
That comprehensive overview really solidifies what exifers are, this new large-scale membrane-bound cellular extrusion. Now, if we connect this to the broader picture, cellular quality control, it immediately sparks the thought, okay, could this be a novel arm of the proteostasis network? So how does the neuron actually decide what to pack into these exifers? Is it selective? And what are the mechanisms behind that sorting, especially for problematic proteins? And how does this complement or maybe even challenge our existing models of proteostasis?
You've absolutely hit on the crucial point there. That selective packaging of harmful material. That's precisely what first strongly connected exifers to the proteostasis network in our minds. We initially observed this, again, through time-lapse microscopy. We were working with aggregating M. Sherry. That's a fluorescent protein known for its tendency to misfold and clump up. And what we saw was, well, striking. This aggregating M. Sherry was clearly being preferentially concentrated into the budding exifers. It wasn't just a random sampling of the cytoplasm. There was a definite enrichment of that problematic protein within these nascent exifer structures. That was our first really strong clue that exifers weren't just indiscriminately ejecting cellular contents.
So not just taking a chunk of cytoplasm, but actively grabbing the bad stuff.
Exactly, it looked targeted. So, to really test this idea of selective expulsion, we moved into neurotoxic protein models, specifically ones relevant to human neurodegenerative diseases, which is obviously a major interest. We used a C. elegans model for Huntington's disease. In this model, neurons express a polyglutamine expansion protein, Hunting Q128CFP. This protein is known to be toxic. It forms aggregates, causes neuronal dysfunction, and we found that these neurons could effectively concentrate and then extrude this specific aggregating Q128CFP protein within exifers. And crucially, the strains expressing the toxic Q128 protein. They produce significantly more exifers compared to control strains expressing a non-toxic Q19 version or those without the polyQ expansion.
So the presence of the toxic protein itself triggers more exifer formation.
Precisely. We even tracked individual neurons over time and found that a high aggregate load in a neuron on day 1 was a strong predictor of increased exifer production by that same neuron on day 2. We saw something similar in an Alzheimer's disease model too. We looked at Azer neurons expressing the human amyloid beta fragment, AO142, which forms amyloid plaques. These neurons also showed a marked increase in exifer numbers. So this really linked exifer production directly to the presence of known neurotoxic protein aggregates.
Okay, that's strong correlative evidence. But how did you show the sorting was truly selective? That it wasn't just that aggregate-filled cells make more blebs?
Right. Excellent question. To nail down the selectivity, we designed what I think was a rather elegant experiment. We co-expressed 2 different fluorescent proteins in the same neurons. One was our aggregation-prone M. Sherry, and the other was a non-aggregating, well-behaved GFP. Our hypothesis was simple. If sorting was truly selective for the bad stuff, the aggregation-prone M. Sherry should be enriched in the exifers, while the good GFP should preferentially remain back in the soma. And that is exactly what we found. We carefully quantified the fluorescence in the exifer versus the soma. In 22 out of 23 exifers we analyzed, the M. Sherry signal was consistently significantly higher relative to GFP inside the exifer compared to the ratio back in the cell body. Meanwhile, GFP remained relatively higher in the soma. This wasn't just a subtle trend, it was a robust, quantifiable difference. It strongly argues that the neuron isn't just randomly blubbing off cytoplasm. It seems to be actively identifying, concentrating, and sorting these deleterious protein materials for export via exogenesis.
That's very convincing. It really points to a specific sorting machinery at play.
Exactly. And this specific sorting mechanism offers, I think, a unique complement to our existing understanding of proteostasis. Think of it like this. The proteasome and autophagy, they're like your internal meticulous recycling crew, right? They're incredibly efficient for routine maintenance, breaking down individual damaged proteins or maybe small organelles, recycling the molecular components. But imagine if your house suddenly filled up with, I don't know, a ton of old, heavy, potentially toxic furniture that's really hard to disassemble like a massive protein aggregate. Exifers are like calling in a specialized service that literally picks up entire rooms full of that problematic furniture, these large aggregates, maybe even damaged organelles as we found later, and hauls them out whole.
Bulk trash removal, not just molecular recycling.
Precisely. It's a rapid, high-capacity external removal system.
Yeah.
Fundamentally different from the intracellular digestion pathways we've primarily focused on for decades. This allows the neuron to deal with burdens that might otherwise overwhelm its internal, more granular systems. So our next step, naturally, was to figure out what actually triggers exofer production. If they really are part of the proteostasis network, then stressing that network should enhance their production, right?
Makes sense. Overwhelm the other systems, and this one kicks in.
That's what we hypothesized, and that's exactly what our experiments revealed. When we impaired HSF1, that's a core proteostasis transcription factor, essential for regulating chaperone expression, we used an HSF1 Cy441 mutant background. We observed a remarkable, something like 6-fold increase in exofer production. A huge jump. Yeah. So compromising chaperone function clearly ramps up exofergenesis. We also pharmacologically inhibited autophagy using SPOT1, a known inhibitor, and we used RNAi knockdown for key autophagy genes like LGG1 and DSSBT11. In all these cases, interfering with autophagy, exofer incidence significantly increased. Similarly, when we inhibited the proteasome, the cell's main protein degradation machine using MG132 exofer production went up again. The fact that disrupting multiple, distinct, well-established pillars of the proteostasis network, chaperones, autophagy, and the proteasome, all led to increased exofer extrusion. Well, that strongly suggests that exofers are an integral component of this network. They seem to function as a crucial backup, or maybe an alternative response, perhaps kicking in when the cell's traditional proteostasis mechanisms get overwhelmed by mounting intracellular proteotoxicity. So rather than challenging our understanding, it really just adds this new, very significant, large-scale clearing mechanism to the known cellular arsenal for protein quality control.
That's a really profound expansion of the proteostasis concept, moving beyond just internal degradation to this, bulk disposal system. Okay, so that covers the protein side. Now let's turn our attention to another crucial aspect of cellular health, organelles. We're so used to thinking about mitophagy as the primary way cells deal with damaged mitochondria, and maybe autophagy for other bits. To see entire organelles being physically jettisoned, especially, as you hinted, oxidized ones, that's quite revolutionary. Could you elaborate on how this mitochondrial expulsion works, maybe in conjunction with or as an alternative to traditional mitophagy? And what does this mean for how we think about mitochondrial quality control generally?
Yeah, this is truly one of the most exciting and perhaps unexpected aspects of the whole discovery. It really broadens our view of cellular waste management beyond just misfolded proteins to actual whole organelles. Given the sheer volume of exofers, we initially hypothesized they might encapsulate more than just protein aggregates. Maybe whole organelles could fit inside, and indeed that's what we found. Both lysosomes, which we show in the extended data, and importantly, mitochondria can be extruded within exofers. To confirm the presence of mitochondria, we use mitochondrially localized GFP reporters. And when we observed these budding and dissociated exofers, we consistently saw typical mitochondrial shapes inside, punctate or sometimes filamentous morphology. This confirmed they were actual organelles being packaged, not just fragments or general cytoplasmic stuff. For graduate students listening, this is a really profound shift in thinking, right? How cells, especially high-energy neurons, maintain the quality of their crucial powerhouses. It suggests a much more aggressive, large-scale removal option than we previously appreciated.
Okay, so mitochondria can definitely get packaged, but which mitochondria? Was it random, or was there, again, that element of selectivity for damaged ones?
That was the next critical question, exactly. Quality control implies selectivity. So to investigate this quality aspect, we employed MITR-RO-GFP. That's a genetically encoded fluorescent reporter that's sensitive to the oxidative environment within the mitochondria. It basically reports on their redox state. And what we discovered was, frankly, crucial. The mitochondria that were encapsulated within the exofers, they displayed a significantly higher oxidized state compared to the mitochondria that remained back within the soma of the very same neuron.
Direct evidence of damage or stress.
Direct quantitative evidence. It was a clear indication of damage, stress, or dysfunction. We further corroborated this finding using another reporter called mitotimer, which also functions as an oxidation-sensitive reporter. It also showed a proportionally more oxidized signal in exofers. This strongly suggested that older or more damaged, more oxidized mitochondria were being preferentially targeted for expulsion. And to really test this causal link, we treated animals with juglone. That's a compound well known to increase reactive oxygen species, ROS production, and cause significant mitochondrial impairment. And just as we predicted, this juglone treatment led to a significantly higher number of exofers that contain mitochondria. It reinforces that direct link. Mitochondrial stress drives their expulsion via exofers. It's like the neuron is actively identifying and offloading its compromised powerhouses.
That's remarkable. And did you see similar links if you interfered with known mitochondrial quality control pathways like you did for proteostasis?
Yes, absolutely. Just as we linked exofer production to general proteotoxic stress, We explored genetic triggers specifically related to mitochondrial impairment. We found that genetically impairing mitochondrial quality control, for example, using RNAi knockdown of key mitophagy genes like DCT1, which is BNIP3 homolog, or PINK1 and PDR1, which, as many listeners will know, are homologs of human Parkinson's disease genes, PNK1 and Parkin, as well as knocking down genes involved in the mitochondrial unfolded protein response, like OBL5. All of these interventions resulted in a marked increase in exogenesis. So this data really firmly establishes exogenesis as a previously unappreciated mechanism of mitochondrial homeostasis. It suggests that while mitophagy is undoubtedly a vital internal pathway for degrading damaged mitochondria within the cell, exogenesis provides this alternative, potentially larger-scale, expulsion mechanism. This could be particularly important, perhaps, when the internal degradation pathways like mitophagy are overwhelmed, or maybe when mitochondria are so severely compromised or maybe too large or complex for efficient internal processing. It really expands our conceptual framework for mitochondrial quality control. It suggests this continuum from precise internal degradation via mitophagy to this external bulk jettisoning via exophores. It's like the cell has both a scalpel and a sledgehammer for dealing with mitochondrial problems.
A scalpel and a sledgehammer. I like that analogy. This eject button for damaged mitochondria is really quite captivating. Okay, so we've established exophores get rid of both bad proteins and damaged organelles. The next absolutely critical question is, does this actually help the neuron? What's the direct evidence that these exophore-producing neurons aren't just, surviving, but actually performing better? And also thinking about the temporal aspect you mentioned earlier, that bimodal distribution. For a graduate student maybe looking at aging or neurodegeneration, how might understanding that pattern inform their experimental design, maybe their choice of developmental stages for study?
Right, the neuroprotective aspect.
Yeah.
That's frankly what elevates this discovery from just a fascinating cellular curiosity to a mechanism with potentially profound therapeutic implications. We absolutely had to determine if this dramatic expulsion was actually beneficial or if it was merely a sign of severe cell distress, maybe a prelude to death. And the evidence, thankfully, strongly points to a direct functional benefit. First off, through hundreds, literally hundreds, of meticulous longitudinal observations, watching individual neurons over time, we never observed neuronal loss after exophore production.
That's critical. They don't die afterwards.
Absolutely critical. Exophores have a distinct biogenesis from apoptotic bodies, as we discussed. And the soma, the main cell body of an exophore-producing neuron, maintains completely normal ultrastructural features when examined by electron microscopy. This clearly distinguishes exophoregenesis from programmed cell death. These aren't dying cells. They're cells actively doing something. Second, and this is perhaps the most compelling piece of evidence, we directly linked exophore production to enhanced neuronal function. We went back to our Huntington's model, the Q128 CFP worms. These worms are prone to progressive impairment of anterior touch sensation as they age. Their touch neurons just don't respond as well over time. You can measure this decline quite clearly by adult day 4, sometimes seeing as much as a 40–50% reduction in their response frequency to a gentle touch. So to ensure impartiality, we ran blinded behavioral assays. The person doing the touch test didn't know which worms had produced exophores and which hadn't. And what we found was really remarkable. Those proteotoxically stressed neurons that had produced an ALMR exophore earlier, say by adult day 2, exhibited significantly better anterior touch sensitivity later on adult day 4, compared to their stressed counterparts that had not produced an exophore.
Wow, so making an exophore actually rescued function.
It significantly improved it, yes. It wasn't just a slight trend. It was a robust, statistically significant improvement. On average, the exophore-producing neurons only showed about a 10–15% reduction in touch sensitivity compared to the 40–50% drop in those that didn't make exophores. This provides direct functional evidence that clearing out this toxic material via exophores substantially improves the neuron's health and performance. It allows it to maintain its critical function even in the face of this accumulating proteotoxic stress. For anyone studying neuronal aging or resilience, this isn't just some cellular quirk. It's direct evidence of a functional, beneficial, protective mechanism.
That's incredibly powerful data, linking the cellular event directly to improved function.
Exactly. And to further solidify this, we also took a genetic approach. We wanted to see what happens if you inhibit exophoregenesis. We identified 2 genes, POD1 and STB8. These are polarity genes as being required for exophore production in adult C. elegans. When we performed adult-stage RNAi knockdown of these genes, we saw 2 critical effects. First, as expected, we observed a significant decrease in exophore detection. But crucially, the knockdown also led to impaired midlife touch sensitivity. Now, we always have to acknowledge that RNAi interventions can sometimes have broader pleiotropic effects on general neuronal function. But taking all the evidence together, seeing enhanced performance when exophores are produced and impaired performance when they're blocked, it really strongly supports the idea that exophore production is a beneficial neuroprotective response. It's actively helping neurons maintain their functional integrity.
That makes a very strong case for the protective role. Now, about that bimodal distribution you mentioned.
Yeah.
Peak in young adulthood, dip, then another peak later in life. What are the implications of that for researchers?
Yes, the bimodal distribution. It's a really fascinating detail, and I think it has significant practical implications for anyone working in the lab, especially studying aging. We observed exophore production follows this striking pattern over the C. elegans adult lifespan. There's this distinct peak in young adulthood, specifically around adult days A2 to A3. Then production dips quite significantly in midlife, roughly between adult days A4 and A8, but then, interestingly, exophore production reappears and increases again in older animals, typically around A10 to A11. Now, how do we interpret this? We have a couple of ideas. The early peak, the one in young adulthood, might serve as a kind of proteostasis reset right after development finishes.
Like a spring cleaning.
Exactly. A sort of house cleaning of neuronal garbage that might have been generated during the rapid growth and maturation phases. It could be a proactive maintenance mechanism to ensure optimal function as the animal enters its prime reproductive period. The later peak, though, the one in older animals, we propose reflects something different. Age-associated decline in the robustness of internal proteostasis mechanisms. As the animals age, their internal systems autophagy, proteasome function likely become less efficient, maybe overwhelmed, and exophore production then ramps up again, perhaps as a necessary, albeit maybe less efficient, last-ditch response to mounting cellular burdens that the cell just can't handle internally anymore. For graduate students, this bimodal pattern is, I think, a critical consideration for experimental design. It tells you that if you're studying cellular quality control or neurodegeneration or even just basic neuronal function, your choice of developmental stage will profoundly influence the basal level of exophore activity you observe. Targeting the early peak might reveal mechanisms of proactive maintenance and healthy aging. Studying the late peak could illuminate the cellular responses to severe age-related decline and chronic stress. It also hints, perhaps, that interventions aimed at modulating exophore production might have different effects or be most effective at specific life stages. Maybe you could boost the early peak to prevent future problems or enhance the late peak to mitigate ongoing damage. It adds a whole temporal dimension to consider.
That temporal mapping definitely adds another layer of sophistication. But it also raises a crucial question, one that often comes up in cell biology, particularly when we talk about material being transferred out of a cell. The fact that some of this exophore content, as you found, can potentially transit to distant cells rather than being completely degraded locally, well, that opens up a whole new set of implications, doesn't it? Does this imply a potential for, say, pathology spread if the transfer material isn't completely neutralized? Or is it primarily a highly efficient way to offload difficult-to-degrade waste onto specialized scavenger cells? What are the broader implications if this clearance process itself becomes dysfunctional?
This is indeed a critical aspect, and it really points to the complex, potentially dual nature of this mechanism that we have to carefully consider. We observed, as you said, that the fluorescence within helicocephalus diminishes over time, typically over a period of 1 to 12 hours. This suggested that their contents are either being degraded, perhaps by surrounding tissues, or maybe dispersed into the body cavity. Our initial electron microscopy studies, combined with just the basic anatomy of C. elegans, supported the idea that the surrounding hypodermis, that's the skin cell layer, which completely encloses the touch neurons, might mediate the degradation of at least some exophore contents. The hypodermis is known to have degradative capabilities.
So a local cleanup crew.
Potentially, yes. A local, efficient disposal system right there. To investigate this further, we looked into the role of specific engulfment genes, genes known to be involved in clearing away dead cells. We found that disrupting C. elegans apoptotic engulfment genes, specifically SED1, SED6, and SED7, led to an interesting phenotype. An increase in multiple exophores accumulating, sort of stuck associated with the neurons. They weren't being cleared properly. This implies that these genes, or at least the pathways they represent, are involved in exophore clearance, likely by that surrounding hypodermis. However, and this is a crucial distinction we had to distinguish this from classical apoptotic corpse removal, we performed careful experiments using an annexin VGFP reporter. Annexin V binds to phosphatidylserine, or PS, which is the classic eat me signal displayed on the surface of apoptotic cells, and we did not detect PS on the exophore surface.
Okay, so it's not using the standard apoptotic clearance signal.
Exactly. This suggests a fundamentally different clearance mechanism than typical apoptosis, even though it seems to involve some components of the same CD pathway, which is fascinating in itself and points towards perhaps a novel recognition system specifically for exophore removal. But then came the more surprising and perhaps, as you say, potentially concerning finding: the evidence for transcellular transfer to distant cells. We observed that fluorescent M-Sherry protein, which was originally expressed only within the touch neurons, or fluorescent DI dye that we loaded specifically into dye-filling neurons. This fluorescence later appeared in distant scavenger cells called colomocytes. Now, these colomocytes are specialized phagocytic cells. They just float around in the pseudocollomic fluid, which is the body cavity of C. elegans, and they have no direct connection to the outside world or necessarily to the neurons themselves. This meant the material must have traveled through this body fluid after being released from the neuron's vicinity.
So it gets released, floats around, and then gets picked up by these scavenger cells elsewhere.
That's what it looked like. To confirm this, we blocked the colomocytes' uptake capacity. We used a cup4 mutation, which impairs their ability to engulf extracellular particles. And just as we predicted, doing this caused fluorescent particles, presumably derived from exophores, to accumulate outside the neurons, likely within that pseudocollomic fluid. They weren't being cleared by the scavenger cells anymore. So this reveals that exophores can indeed mediate the transfer of neuronal materials to remote cells. Now, on one hand, as you suggested, this could be a highly efficient way to offload difficult-to-degrade waste to specialized scavenger cells like colomocytes. It provides a robust, long-range disposal system for materials maybe the local hypodermis can't fully handle, sort of a communal waste management system, distributing the burden. On the other hand, yes, the potential for pathology spread is a serious consideration we absolutely have to keep in mind. If the transferred material, particularly things like misfolded proteins or maybe even damaged organelles, isn't completely harmless, if it can retain some toxic properties, then this pathway, especially if it becomes dysfunctional, could inadvertently contribute to the propagation of pathology. This is obviously incredibly pertinent in the context of human neurodegenerative diseases, where we know aggregation-prone proteins like tau or alpha-synuclein can spread between cells, contributing to disease progression. The implications are really profound. Understanding the precise mechanisms of this transfer and the subsequent clearance or fate of the material is absolutely essential. We need to evaluate whether enhancing exophore production is likely to be a universally beneficial therapeutic strategy, or if, perhaps under certain conditions, it could potentially exacerbate disease spread, particularly when we start thinking about translating this to complex mammalian systems.
That dual potential efficient disposal versus dangerous spread really underscores the complexity here and the critical importance of understanding this pathway inside and out. Okay, so connecting this now to the bigger picture, particularly those human neurodegenerative diseases you've mentioned, this research clearly suggests that manipulating exophorogenesis could be a genuinely novel, potentially powerful target for intervention. So for our graduate student listeners, maybe thinking about their next project or their future careers, what do you see as some of the most exciting unanswered questions arising directly from this work that they might pursue? What specific areas of research do you think could have the most immediate impact, especially given the hints that similar phenomena might be conserved across species?
Right, when we connect this to the bigger picture, the potential for exophores, or something very much like them, to be a conserved pathway across species, that's incredibly exciting and holds immense translational promise. There's already, as many listeners will know, significant enthusiasm in the neurodegenerative disease field about findings that mammalian neurons can extrude conformational disease proteins, things implicated in Alzheimer's, Parkinson's, prion diseases. Our discovery in C. elegans provides a newly identified large-scale mechanism that could underlie or at least contribute to this kind of transcellular material transfer. And there are these tantalizing morphological and even functional parallels cropping up. For instance, in the mammalian systems, aggregated polyQ huntingtin, just like the protein we studied, can transfer between neurons via structures called tunneling nanotubes. These nanotubes bear a really striking resemblance morphologically to those thin, thread-like connections we often observed between the C. elegans somas and their budding exophores. It suggests there might be a potentially shared biogenesis mechanism there.
Interesting parallel.
And in Drosophila, the fruit fly neuronal polyQ protein can transfer to glial cells. And this transfer requires a protein called Draper. Traper is the fly homologue of C. elegans CED1, which, as we just discussed, we identified as being involved in exophore clearance in the worm.
Another conserved link.
Exactly. And perhaps most strikingly, thinking about the mitochondrial quality control aspect, we see a direct mammalian counterpart that really lends strong support to the idea of conservation. Mouse retinal ganglion cells, work from other labs has shown, are known to extrude mitochondria into neighboring astrocytes, presumably for degradation by the astrocytes. And the morphology of these expelled mitochondrial structures in mammals appears an intriguing similarity to the C. elegans exophores we described. So seeing these kinds of observations across such diverse organisms and cell types from nematodes to flies to mammals leads us to strongly speculate that exophore production, or at least a highly analogous process, might be a deeply conserved pathway, a fundamental mechanism for transferring toxic or unwanted contents out of multiple cell types, not just neurons. For graduate students, this absolutely opens up a wealth of fundamental and translational unanswered questions that are just ripe for exploration. That dual nature of the transfer of the potential for beneficial clearance versus pathological spread, that's clearly a major area needing intense research. First, just on the basic mechanistic side. How exactly is specific cargo selected for packaging into an exophore? Is there a specific receptor, maybe, that recognizes misfolded proteins or damaged mitochondria and targets them to the budding site? What's the specific molecular machinery, the signaling pathways that drive that initial balloon-like extrusion from the soma? And then the precise pinching off, we need to find those molecules. And critically, on the clearance side, what are the specific signals on the exophore surface that guide its fate if it's not the standard PS eat me signal? How do surrounding scavenger cells, whether it's the hypodermis or colonomocytes in the worm, or maybe microglia and astrocytes in mammals, recognize and degrade these contents? Uncovering these molecular players is absolutely paramount.
Finding the specific genes and proteins involved.
Exactly. And then, from a therapeutic perspective, the most immediate impact could come from investigating whether actively enhancing exophoregenesis could be a viable and safe strategy to boost neuronal health in models of human neurodegenerative conditions. Could we identify specific drugs or maybe genetic methods to selectively increase beneficial exophore production, especially, say, in older animals or in disease models where we know proteostasis is faltering? Conversely, and this is just as important, how do we prevent the unintended spread of potentially toxic materials if this process goes awry in the human brain? Can we perhaps block the release or enhance the capture and degradation by specific scavenger cells? Identifying the full spectrum of molecular players involved in formation, cargo loading, releasing, clearance will undoubtedly lead to novel diagnostic tools and completely new targets for therapeutic intervention. This work, we hope, doesn't just add a new chapter to the cell biology textbooks. It really invites a new generation of researchers to explore this fundamental, yet formerly unrecognized branch of cellular quality control, one with, we believe, immense therapeutic potential. The first labs to really decipher these mechanisms in detail, especially in mammalian systems, they're going to make a huge mark on the field.
What an absolutely incredible deep dive into the world of exophores. Truly fascinating. We've seen how C. elegans neurons actively eject protein aggregates and damaged mitochondria, a fundamental large-scale mechanism of cellular maintenance that, as you've shown, significantly boosts neuronal health and function. From that serendipitous initial discovery, watching those fluorescent blobs, to the detailed analysis of cargo selection, the clear neuroprotective benefits, and unraveling the complex journey and fate of their contents, it's abundantly clear this is a pivotal finding. It really reshapes our understanding of cellular quality control. And just imagine for a moment a future where we could strategically enhance this natural garbage disposal system in our own neurons, perhaps to prevent or even reverse the accumulation of toxic proteins and damaged organelles that drive so many devastating diseases. Or, conversely, a future where we could specifically block the unintended spread of toxic materials if this process goes awry in conditions like Alzheimer's or Parkinson's. The discovery of exophores doesn't just add a new page or chapter to cell biology. It really feels like it opens an entirely new book of therapeutic possibilities. It offers a genuinely fresh perspective on the challenging landscape of neurodegenerative disease research. The profound question now becomes, how do we harness this knowledge safely and effectively? How do we translate these elegant findings from the humble nematode C. elegans into meaningful, tangible interventions for human health? Thank you so much for sharing your invaluable insights with us today, for walking us through your team's pioneering research, and really for opening up such a vibrant and exciting new frontier in cellular biology. For all of you listening, especially those of you deep in biomedical research yourselves, we sincerely hope this deep dive has sparked your own curiosity, maybe given you some new ideas, and encouraged you to consider the profound implications of this work for your own studies and your fundamental understanding of neuronal health and aging.